Hi, my name is Melina Giakoumis, and I'm a PhD candidate in biology here at CUNY. I study a couple of species of sea star in the genus Asterias. Both are found in the North Atlantic Ocean. This group has a really interesting history. The ancestral Asterias species was exclusively Pacific, but about 3.5 million years ago, the Bering Strait opened between Alaska and Siberia, and Asterias made its way to the Atlantic. Then, shortly after arriving, it split into two species. One found only on the European side of the Atlantic, and one found on the North American side. The two were isolated from each other for a long time, before the European species colonized North America. Now, the two overlap on a small portion of the east coast of North America. For some time, scientists have thought that hybridization might be going on between the two Atlantic species, but no genetic evidence of this happening in the wild has ever been uncovered. Hybridization can be really informative about how a species reacts to changes in climate and where it may be in the future. So I was really interested in seeing if this hybridization was actually happening. The area where two interbreeding species meet is what's known as a hybrid zone. A handful of people have studied this group before me, and many of them estimated where this hybrid zone might be. As you can see, there isn't a ton of overlap between these range estimates. So to get started, I needed to get out on the seashore and do some sampling. So this is what sea star sampling looks like. I spent a couple of summers driving up and down the east coast of North America, from Florida to Canada, looking for sea stars. Sometimes I would snorkel for samples, and sometimes I would wade out at low tide and poke around. These trips also involved using my car as a mobile lab, and I can tell you it's just impossible to fully remove the smell of sea star once it's set in. Once the sampling was done, I looked for genetic evidence of hybridization between the two species. Now, what would genetic evidence of hybridization look like anyway? Let's say we have two groups of sea stars. They're separated by this gray barrier for a long time. These colored bars represent what their genomes would look like. They're completely different from each other because they've been separated for so long. One species is represented by green genomes, and the other species is represented by orange genomes. Now, what happens when the barrier goes away? If the different species are closely related enough, they may be able to breed, in which case, in the area where they overlap, you would see some mixed genomes, which shows ancestry from each species. But in the areas where they don't overlap, you'll see individuals with no mixed genetic ancestry. So once I sequenced my first set of samples, it turns out there was strong evidence of hybridization. This is the same type of representation as in the previous slide, but overlaid on a map of sampling locations. Each vertical bar is representing an individual sea star, and each color represents ancestry from one of the two species. As you can see from this figure, the northern North American samples have similar genetics to the European samples. They're all orange. And the southern samples from North America are also very similar to each other. They're all green. But hybridization begins to occur in northern Massachusetts and continues through much of Maine. Since I created this figure, I've gotten some brand new data, which suggests there may even be some hybridization happening as far north as Nova Scotia, noted here by the black arrow. So my next steps are to figure out what this hybridization means for the future. I'm interested to know, is hybridization new, or has this been going on for a long time? Will these species move as the climate changes, and will that affect how often they hybridize? And what does this mean for the rest of the community? I'm really excited to continue this research and answer some of these questions in the next couple of years. Thanks so much for tuning in. I'm really excited to chat with all of you and hear your questions.